Hello, my dear students. I'm Dr. Vaishaly Bharambe. I've been teaching anatomy for the last 25 years and I love it. This lecture is a part of my embryology lecture series titled as Fertilization Part 2. Take a look. The female produces the secondary oocyte by process of oogenesis. The male produces sperms by process of spermatogenesis. Both secondary oocyte and the sperms travel to the ampulla fallopian tube where the sperm may penetrate the oocyte to bring about fertilization and production of an embryo. This whole process, is coming under the category of fertilization and factors affecting fertilization. Today's lecture is about contact and fusion of gametes, that is fertilization part 2. In my earlier lecture, I have already discussed how the sperms and the oocyte have transported themselves to the ampulla or fallopian tube. Today, we will see whether they fuse or not. So, for understanding contact and fusion of gametes, let's first understand the structure of the secondary oocyte and spermatozoon. Then after that, I will talk about how the sperm is able to penetrate into the oocyte. Then I will talk about changes in the oocyte after the sperm has penetrated into it. So what is our lecture going to be like? First, we'll understand structure of oocyte, structure of sperm in brief. Then we'll talk about how the sperm manages to enter into the oocyte. Then we'll talk about changes in the oocyte after the sperm has entered into it. Ready? Let's begin. First, structure of secondary oocyte and structure of spermatozoon. Right. So here you are seeing a secondary oocyte. It is protected by 3 layers. What are the 3 layers? Take a look. A Vitelline membrane which immediately surrounds your secondary oocyte. A Zona Pellucida, which is a thick layer surrounding that, and a corona radiata bunch of cells along which the secondary oocyte was ovulated out from the ovary. The Vitelline membrane and Zona Pellucida are separated by perivitelline space containing the first polar body. Basics. You can see that along the outer edge of the secondary oocyte, there are a series of cortical granules present. So here, this is an enhanced high magnification image. Here you are seeing a secondary oocyte. You can see that it contains 22 plus X number of chromosomes, the chromatids of which are suspended on the meiotic spindle. Why is it like that? Because the whole cell multiplication, division was arrested in metaphase of meiosis 2. If you recall, if you've studied oogenesis, that during meiosis 2, the secondary oocyte division was arrested in the metaphase of meiosis 2. So there is no nucleus. You can see a spindle, you can see the cells, the chromosomes suspended on the spindle. The secondary oocyte is surrounded by perivitelline space. This is the Zona Pellucida surrounding it, and these are the corona radiata cells. So remember, all this has to be traversed by the sperm if it has to enter into the secondary oocyte. Meanwhile, what is the spermatozoon like? What is its power? So this is the spermatozoon containing the head, containing 22 plus X or 22 plus Y chromosomes. Its head end is covered by an acrosomal cap containing the following enzymes. Let me read them. Hyaluronidase, acrosome, acid phosphatase, etc. Ma'am, how do we remember this? Isn't it? There are so many enzymes. I'll tell you how to remember. Use the mnemonic HAPPEN. HAAPEN. Sperms make things happen? Yes? Sounds quite awful, but that's the way it is. H for hyaluronidase, A for acrosome, A for acid phosphatase, P for protease, E for esterase, N for neuraminidase. Now you can remember almost all the enzymes. Use your mnemonics. Don't just think the problem is big. Solve the problem. Right. So here's the middle piece just behind the head, and that's the tail. So what do we have today? We've got an oocyte which is sitting in the secondary oocyte sitting in the ampulla fallopian tube, and we've got 300 to 500 sperms which have managed to make their way into the ampulla of fallopian tube, and they are trying to enter into the secondary oocyte. If they want to enter in, they have to pass through 3 layers. What are the 3 layers? One last time. Vitelline membrane, Zona pellucida, corona radiata. Say it once more. Vitelline membrane, Zona pellucida, corona radiata. If the sperms manage to pass through this, then only it can enter into secondary oocyte. Otherwise, it cannot. Okay? So, that's the sperms challenge. Okay? Students, if you're enjoying this lecture, do like and subscribe to my channel. I would love it if you pass some comments and allowed me to understand whether these lectures are of use to you or not. Okay? Right. Let's continue. So, we've discussed the structure of oocyte, what are the layers protecting it. We've discussed the structure of the sperm in short. Now, what's our challenge? Our challenge is to understand how the sperm is going to pass through to reach into the secondary oocyte. That's the next section of our class. Aim is for spermatozoon to penetrate 3 layers surrounding the secondary oocyte. Which 3 layers? Vitelline membrane, Zona pellucida, corona radiata. Okay. But first, the sperm must prepare itself for the journey. What is that preparation called? It's called as capacitation. Only a capacitated sperm is capable of passing through the corona radiata and entering into secondary oocyte. It's a very common short note. Do prepare this well. What is capacitation? Capacitation is the conditioning of spermatozoon in the female genital tract. Where does it occur? It occurs in the uterine tube. How much time does it take? It takes about 7 hours. And what is involved? Exactly hota kya hai ma'am capacitation mein? What happens in capacitation? Basically, there's an epithelial interaction between the mucosal folds of the fallopian tube and the spermatozoon itself. Let's take a look to understand what happens. Here's a spermatozoon covered by glycoprotein and semi plasma protein. See the yellow things, okay? What happens in capacitation is that these are lost. You can see that they are lost. And suddenly, the molecules that are covering the head end of the sperm, which are capable of binding Zona pellucida, are now exposed. Till now, they were covered by these plasma proteins and glycoproteins. When you lose the plasma proteins and glycoproteins, okay, the surface molecules are exposed and they are now capable of binding with Zona pellucida. Understood? So, this process, at the end of this process, the sperm is called as a capacitated sperm. Okay? And again, I'll say it one last time. Only a capacitated sperm is capable of penetrating the corona radiata to reach the Zona pellucida. So, how will you write your short note? Revise quickly. What is capacitation? Where does it take place? How much time does it take? What is lost in capacitation? Why do you want to lose the glycoproteins and plasma proteins by process of capacitation? Prepare this note well. It's asked pretty often. Right. So, what next? Okay. The capacitated sperm is now ready to pass between the corona radiata cells. See how it passes. It's easily passing. It's having no problem. Because it is capacitated, it can pass through corona radiata. Think. Sperm has undergone capacitation. Sperm has started its journey by passing between the corona radiata cells. Now what happens? When it comes in contact with corona radiata cells, the sperm surface undergoes some changes. I'm not going to read them. I'll show you the changes. This is the sperm head. This is the nucleus of sperm head. This is the plasma membrane of the head of the sperm, and this is the outer layer of acrosomal cap. See the fluorescent green is your acrosomal cap. You remember what are the contents of acrosomal cap? HAAPEN, right? So, now what happens? When it comes in contact with the corona radiata, something changes in the sperm. This plasma membrane fuses with the outer layer of acrosomal cap. Can you see how tiny little holes have developed and the two membranes have fused? Now acrosomal cap is suddenly open and it starts to release its enzymes. Which enzymes? Hyaluronidase, acid phosphatase, acrosome, and so on. Okay, use the mnemonic. And this is how these enzymes are released, and the process is called acrosome reaction. Okay, these enzymes are powerful, students. They are powerful enzymes. Let's see what happens now. This is what is left of the head end of the sperm. Okay, so here you're seeing how the enzymes are being released. Okay, by acrosome reaction, you can see the head end of the sperm has become porous and all those enzymes are flowing out. Now what happens is that the head end of the sperm binds with a receptor called ZP3. See, the Zona Pellucida is made up of proteins such as ZP1, ZP2, ZP3, out of which the head of the sperm is capable of binding ZP3. When it binds with ZP3, you can see that happening there. The enzymes that are released begin to digest the Zona Pellucida now. See point number 4, you can see the enzymes here which have been released and they have started to digest the zona immediately around that sperm. So as the zona gets digested, sperm makes its way forward till it finds itself in the perivitelline space. See point number 5. So it has passed through the Zona Pellucida and is now lying, the head end is lying in the perivitelline space. 2 layers are already done. The zona, the corona radiata is done, the Zona Pellucida is done, but Vitelline membrane is a challenge. Okay, how do you cross a membrane? Okay, what the sperm does is it just fuses with the membrane. It decides, I will give away myself to the oocyte. It just fuses. The plasma membrane of the sperm fuses with the Vitelline membrane. So the sperm now becomes open. And the head end of the sperm along with the tail enter into the secondary oocyte. Cytoplasm remains behind. Only the head containing the nucleus and the tail enter into the oocyte. Are you seeing that? Okay. So what have we done? Did you notice the sperm has managed to enter into the secondary oocyte? Good for you. Okay. How did it do that? It underwent capacitation. It penetrated corona radiata. It underwent acrosome reaction. The 2 plasma membranes fused, and finally sperm is sitting pretty in secondary oocyte. Now the job is done. My job is done. Let secondary oocyte manage now. Okay? Abhi kaam hota hai secondary oocyte ka. Let's see what is going to happen now. So, the moment this is the sperm that you can see, and this is the secondary oocyte in which the sperm is sitting pretty. Okay, secondary oocyte now feels the presence of a sperm inside it, which causes a calcium wave to just shoot through the secondary oocyte. The whole thing is now feeling calcium waves passing through the cell. And because of this calcium wave, some changes take place in the secondary oocyte. Which changes? They are called as Zona reaction, resumption of meiosis 2, activation of this oocyte to complete cell division. I'll explain it to you. We don't have to mug up. Okay. So, we've reached a stage when there is a calcium wave in the secondary oocyte. What happens because of it? Because of which these cortical granules that I had shown you earlier, they release their content out. They just burst open and they release their content. And the adjacent Zona Pellucida undergoes some changes. If you can see the changes here, okay, it becomes impermeable to any other sperm. My sperm has entered inside, nobody else can now enter. Let's take a look, okay. So here's all these sperms are trying to enter, but there is already a sperm sitting inside and none of the other sperms are allowed to enter. Okay, this prevents polyspermy. Just imagine that a secondary oocyte receives one sperm and then receives another sperm. So the progeny will have 24 multiplied by 3 number of chromosomes. Not acceptable, isn't it? So this is prevented by this process called Zona reaction. What is it? This process is release of cortical granules, material in the cortical granules, causing the Zona Pellucida to become impermeable to other sperms. A similar impermeability also affects the Vitelline membrane, causing something called Vitelline block. So Zona ne block kar diya, Vitelline ne block kar diya. Zona pellucida has blocked sperms, Vitelline membrane has blocked sperms. Whatever is going to happen only inside the secondary oocyte now. Nobody else can come in and interfere. Okay, so the secondary oocyte is now stimulated by the calcium wave to complete its meiosis. You remember, secondary oocyte was arrested in metaphase of meiosis 2. So it completes that division, resulting in development of a definitive oocyte and a second polar body. Remember, there was only one polar body in the perivitelline space. Now there are two. The nucleus that is now formed, okay, becomes enlarged, puffy, vesicular, and is called as the female pronucleus. Definitive oocyte develops a nucleus which is large, vesicular, and is called as female pronucleus. Question is, what is the sperm doing lying around in secondary oocyte? Kuch kaam nahi, it has nothing to do there? No. Now the sperm gets stimulated to become, the head end becomes large, puffy, and forms the male pronucleus. So there's a female pronucleus and there's a male pronucleus. They come to lie next to each other now. What about the tail? The tail degenerates, students. Tail has got no business inside that secondary oocyte, so the tail degenerates. Now what do we have? We have a definitive oocyte containing a female pronucleus and a male pronucleus. Now what will happen? Now, both the male and female pronuclei containing chromosomes first duplicate their DNA material. All this DNA now gets attached to a mitotic spindle. So there are chromosomes from the male and there are chromosomes from the female parent, and they're all suspended on that single mitotic spindle. Do you realize that this results in diploid number being restored? The diploid number is now restored in that cell. Such a cell with these chromosomes in it suspended on the spindle is called as a zygote. Because there is a spindle, you know that what follows. Like any other mitotic division, in the chromosomes split longitudinally and there is formation of 46 chromatids, each of which moves to the pole of the spindle. By process of telophase, there is cytokinesis and two cells get formed. So here's one cell and here's the other cell. Okay. So there was just a moment when all these nuclear material was together as one cell. And then immediately it goes into cell division and the zygote now reaches the two-cell stage. Okay. You can see that the cells, both the cells are smaller than the parent cell. Why? Because it is still enclosed in zona pellucida, which is not going to expand. So in that space, division means reduction in size of the cell. So the ovum or the definitive oocyte was larger and the daughter cells are smaller. Okay, enclosed within the zona pellucida. There are some notes that I have put in here. I need you to read them, but I will not discuss them. Feel free to call me in case you want to discuss this. So, what have we seen? Let's revise quickly. As I say, images speak a lot. This is the secondary oocyte arrested in the metaphase of second meiotic division. These are the cortical granules. You can see here the sperms are trying to penetrate into the secondary oocyte. You can see here that one victorious sperm has managed to enter in. Now what happens? As it enters, the other sperms are not allowed to enter by process of zona reaction. Meanwhile, the secondary oocyte completes its metaphase and completes the second meiotic division, and there is formation of definitive oocyte and the second polar body. The nucleus of the definitive oocyte becomes enlarged and forms the female pronucleus. The sperm head now forms the male pronucleus. Male and female pronuclei now enter into mitotic division, uniting on a single mitotic spindle, and finally there is the two-cell stage, zygote formed. All this is what we have discussed under the heading of fertilization part 2. Changes in the secondary oocyte One last time, let's revise. When the sperm enters secondary oocyte, a calcium wave is set off. Calcium wave causes the release of cortical granules, resulting in Zona reaction, Vitelline block. Second meiotic division is completed in the secondary oocyte with formation of definitive oocyte and second polar body, and formation of zygote with male and female pronuclei. Finally, cells complete their first mitotic division and development of the two cell stages observed. This entire process is fertilization part 2, contact and fusion of gametes. I suggest the next lecture will be about effects of fertilization and applied anatomy. Do watch that. My strong advice to all of you would be watch lecture 1, which is transportation of gametes, followed by this lecture, followed by effects of fertilization. Watch all 3 to really understand the process of fertilization. Students, if you like the lecture, do like and subscribe to my channel and do share your thoughts on these lectures. I would love to provide you whatever else you need to help you in your journey in education and medicine. Thank you, students. Bye.